so that like when we think you can also specifically come up
Shall we start?
yeah, please.
Yes. Okay. So, few audience here, but I think
Some are there online.
Okay. So I'll start with a brief introduction. I said brief, sometimes always expanded, but I like to keep it brief all the time. How long the session is, Dr. Randy?
Till 1:30 would be fine.
Okay, so as I mentioned, like this is the GAN project, and my title is here. And as you know, I'm the Bapi Pahar, and anytime you can email me, and also that is my US phone number, and the website is there, so you can log in the website and can find out more about me. Though I recently joined Michigan Tech, I was before NIH, so I'll be happy if you are in any future collaboration, future how to say, future endeavors, like exploring PhD program, Michigan Tech, or anywhere else, any project-related things, any discussion on biology, like that is my expert, so I'll be happy to discuss more. Especially my interest is to combine the technology and the biology, where I mentioned initially the biosensors and the tissue scaffolds, nanotechnology, how you can incorporate those new technologies to make a better diagnostic, better vaccines in future. So next slide, please. So just give me some background about me. I'm an immunologist, virologist. I'm an expert in BSL-2. Some of them don't know what is BSL-2. BSL-2 means biosafety level 2, 3, and 4. So there are 4 levels, 1, 2, 3, 4, based on how infectious this disease is, how dangerous this disease is. So they have been categorized by the CDC, 1, 2, 3, 4. So I have worked starting from 2 and 4. As you know, I think this institute has mostly BSL-1 and BSL-2. I don't know, some Randy can explain more. They have some food, biosafety food. If you're using biosafety food, it's mostly BSL-2. And also I'll discuss maybe in a day 5 or so, or 4, some more demarcation about how BSL-2, BSL-4 at a later point. So I have expertise in those organisms. I have expertise in vaccinology, pathogenesis, drug discovery. I have over 85 publications and book chapters. We recently published a book chapter. I will recommend that to read if you're interested in that field. I'm editor of Discovery Medicine, Vaccine Frontiers Immunology. I am a review board of several NIH funding and multiple scientific journals. So just a schematic how I fit in this talk this week. Emerging and re-emerging symptoms to highlight my expertise in different BSL-2 and BSL-4 expertise. So far I have gained during the course of my research area in Tulane as well as in NIH and others. Next slide, please. So this is kind of curriculum. I think you all have those curriculum, but there's maybe some changes. Ups and downs, but basically I'll be covering BSL-2 pathogens, BSL-3 pathogens. Some of them you may not heard it about from me. BSL-4 pathogens, and talk about something what is biosafety and biocontainment. Like we all experienced COVID infections. 2020, I think that is more fresh to us. And recently I saw the newspaper in Times of India. There's a lot of outbreaks happening on flu in New Delhi and Chandigarh and those places. So definitely we should be careful about, because flu is an emerging disease and it always happens. So how you should know a little bit about biosafety and biocontainment if you are really working on the future, or even if a person got infected and saw in the street or in your home or somewhere, how you would try to handle those. And the next day, last day, I talk about the next generation technologies, some of the recent developments, what we are also doing. So I'll give some emphasis on those, how we are doing and how the future, the coming generations are looking in those directions to discover the diagnostics and vaccines. Next slide, please. So, some of the terminologies, I'll be going through throughout this week, you should be familiar with, because I'm talking about health science, not engineering, so it should be familiar with what is endemic, what is epidemic, what is a pandemic, endemic. So endemic means prevalent of human infections, epidemic, highly incidental infection disease. Pandemic means it's everywhere, any country it has been spread on. Endemic means it's only limited to the animals, not in the humans. So when it calls human, it's endemic, okay? And when it is animals, it's zootic, okay? So endemic, panzootic, that kind of. So it is prevalent to all pigs, all cattle, then it's panzootic and that kind of thing. So newly emerging disease, disease recognized in human for the first time. So that is a newly emerging disease, like because HIV, we know it has been first discovered in 1981, and still, we have HIVs, flaring up. We saw mostly in the Asian countries, African countries, it's still prevalent. There are 31 million people die so far after the discovery of the HIV, and still people are dying. There are a lot of countries that don't have drugs and all those. New emerging diseases that have historically infected humans but continue to reappear, like West Nile. Like West Nile, it was earlier in Europe. Now it has came out to the United States. I'll talk about some of them. So it is an emerging disease. Because another thing, it has no vaccines. Then deliberately emerging infectious disease. What we do here, like especially anthrax, probably you heard like fighters and like mass destruction, like Saddam Hussein, you probably heard about this. People thought, okay, they have the mass destruction something, right? Either nuclear bomb or some biological agents. So biological agents, people have used anthrax is one of the major, which have been factors. Probably you've heard, United States, they have like an envelope full of powders and all those things. So those have accidentally emerging infectious disease. So it may be, like transmissible vaccine-derived poliovirus. It's accidentally happened, right? Suppose you are making your vaccines and you have not done proper QC. It has not been inactivated properly. Maybe it has been marketed. So it is kind of accidentally emergency introduced. It is not supposed to be. So next. So there is a list of disease. I think there's a slide got here. So the different years there are a lot of disease outbreak happened. So you can see BCE, the plague of Athens happens. Then recently they have Ebola viruses there, and acute hemorrhagic conjunctivitis, HIV, SARS, CoV-2. Zika virus in 2015. So those are the viruses emerging, and there are a lot of diseases spreading. Next slide. This is also a busy slide. So recent emerging diseases. I'm not going to in details, but I can share the slides later on. You can see those red mark is newly emerging disease. The blue are re-emerging disease, and the black is the deliberately emerging disease. So those are the diseases you can see they are playing up, especially interested in re-emerging disease. So you can see MERS, E. coli, the Nipah virus, which is I'll talk about I think day 3 or so. Enterovirus. So there are a lot of viruses, and you can see the geographic distribution. So, this is the America, this is the Africa, and Asia, and Oceania, and so you can see there are a lot of outbreaks in different countries and all those. So, and definitely you can find out we are living with so many different diseases and so many countries. Even if we travel some other countries, there may be some emerging happen, and we don't know. So that's why always look over travel advice and say, hey, how the condition is there. Is there any outbreak? Is it safe to travel? And what I carry? How to protect myself? Shall I need a vaccine before I go there? So those things we have to be, as a 21st century, we should be careful. You cannot go back like 30 years ago. What is happening? I travel anywhere, but no, we have to be very proactive on those aspects and do it. Next slide, please. Previous slide. Oh, okay, that's what I was looking for. This is for 400. Oh, okay. Yeah, I know. Okay. Thank you. So yeah, thank you for your interest and enthusiasm. And throughout this week, we'll be interacting more. I mean, I really want your feedback. And if you have any questions, please ask. Please raise your hands in between talk or so. Or if you can hold your questions, you can discuss, maybe during lunchtime or after the break or at some point. You can email me also, as I'm here, so you should talk more. Yeah, this is I'm repeating again, my phone number and email, so definitely you should, I need some feedback from you guys. And as putting some pictures of where I live now, the snowfall already started. So, when I go, I think next week, they say it is already freezing temperatures below zero. So, I'm scared about driving the snow, so, but they mostly clean the road. So, okay, so we'll go to the folder, maybe dengue. That's the last. So we talk about dengue. So I think you guys all know, right? Ladies, dengue. You heard about dengue. Always we see, governments, put so much advertisement on the streets or bus and everywhere. So what is the dengue actually? It's not working. Next slide. This remote is not.
Just check that. You check it once again now.
No, it is not working. But it's taking time. I don't know. It is a lapse. Okay, fine. Okay, so dengue is a flaviviral. Well, throughout this talk, I'll be talking a lot of genus, family, all those. No need to remember, okay? But I'm not asking questions on this. I'm not taking any exam, anything. But just to understand, if anybody interested, I think I like to make always my presentation like a full form, right? Not half-hearted or something. So I put the genus, and those are important because there are a lot of families there. So you should, in future, if you want to know, dengue, what is the family, why it is important. That's another class we have to talk about what is genus, family, and all this. But for the purpose of the lecture, I just do those. Sometimes you see envelope, sometimes see icosahedral capsid. No need to remember. We can talk later. But just understand this is a virus. It has a RNA virus or DNA virus. Those are important. If it is a positive strand or negative strand, those are important. But how the structure, I don't care that kind of. I'm an engineering student. I don't care. But yes, you don't care. But you should know. Okay, if somebody say, "Oh, dengue," you got the lecture, but to the families, okay, So it's also for your knowledge to know those things. But I'm not going to question anybody. Okay, but it's family. So this is for your informations. But just in future. The point is the virus with the same genesis. So you can see those are clusters. Those are the phylogenetic tree based on like this Deparseviri virus, Springy virus, flavivirus, and pestivirus. We'll be talking more. The flavivirus, where is the West Nile virus, Japanese encephalitis, a lot of outbreak happening in India. Japanese, it's a yellow fever virus, not too much here. Dengue virus is very much. So they are in the same family. So when I will be talking West Nile, oh, it is similar like dengue. That's the point is, because they are in the same genus, same family, and but they have a similar kind of replication. Replication means how the virus multiplies, how the virus, one virus make two virus, or 3 or 4 millions. So that's the thing. So that's the idea of here to understand. Okay,
Next slide.
it's not going here.
GIAN, can you
Next slide. So, next slide. Yeah. So it's another the structure of the virus. Where virus, as I said, virus cannot be seen in your open eyes, neither with the glass. You need a special kind of microscope, which is electron microscope mostly. Only one virus you can see under regular microscope, that is the pox virus, which is very big. But normally all those viruses, when we're talking this whole week, you cannot see their eyes. That's very, very small. It's a nanometer. So but they are very fancy in the sense they are well organized. Though they are so nanometer small, they have everything they need to replicate. So they have like the RNA genome which can replicate, the membrane protein, envelope protein which cover the virus, protect the virus, their RNA. Because as you know, as a human being, we wear dresses. When it is cold, we have more dresses. When it's hot, we have like a Bermuda and all this. So virus also has to cover their own RNA, their whole body. So they have their envelope is very important. And the envelope is most of the vaccines so far generated based on those envelope proteins. That because those are immunogenic. Immunogenic means that gives more better immune response. So here is the structure, like the virus has the envelope protein, capsid proteins. It has the RNA genome, and the genome is like this. So always the genome has structural proteins and non-structural proteins. In throughout my different virus courses, I have been talking those non-structural, structural. So structural means that gives production of those structural proteins. They are actually present in the virus. Non-structural, they are actually responsible for replication, how the virus replicate from one virus to two. So they are not actually present too much. They are not. Their function is only for replication. But the structural is more important because you can make vaccines or anything based on the structural proteins. There was some studies we did long ago to find out how the non-structural, that's a different topic, but non-structural protein some way is important for better diagnostic, better outcome. We have seen a lot of studies when you are doing HIV, so some, if you generate antibodies, that's non-structural proteins, you have a better prognosis. Prognosis means better outcome. They may live longer on those rather than others. So that is a different thing we can discuss later. Next slide. Somehow this is not, yeah, you can check it.
So down arrow, yes. The down arrow doesn't.
It's not moving easily. So the dengue, so this is the latest report, 2024 to May 25. We can see, and here's the how much percentage infected. You can see major infection is, you can see those African countries, some of the African countries, lot of Europe's, okay, Southern Europe's like Brazil and all those, Cambodia, those things. You can see Malaysia, Thailand, those. India is in between the presence there. So America also has dengue because somehow mosquito went there because it's transmitted to the mosquito. So now it is working. Okay, so this is the dengue cases in India. I find out from the website. You can see all the like 2019 to 2025 so far. I think it's still February. 49,000. So the 49,000 people has been died due to or got infected with this dengue viruses. So this is a replication, as you know, how the virus replicating. So can you put the video, put the YouTube, press on the link and it will open if you are able to get that. It needs YouTube to open. So you cannot see it. Well, they're opening in a different window, right? So you can see the virus is binding to the receptors, virus is encapsulated. So this is the typical pictures, and also that's why I put the YouTube. It will be easy to understand, and as a video format. So virus got entry, endosomes, then endosomal vesicle is very pH dependent, and the virus release the RNA nucleocapsid. So Golgi ER means endoplasmic reticulum. So two things very important in the dish in each cell: endoplasmic reticulum, Golgi apparatus, and nucleus. Endoplasmic reticulum helps to generate more non-structural proteins and all those. Golgi it's kind of an assembly. Like we're making vaccine and then a separate department where all the vaccine assembly happen, like putting labels, make sure that we have enough vaccine, enough dose of the vaccine. So Golgi apparatus, which is in our cells, do that kind of thing. When the virus produces RNA, proteins, everything, Golgi makes everything assembled. So intact virus produces that. And then it releases from the cells. Endoplasmic reticulum, it is a translation where is the RNA on all those non-structural proteins are producing, forming here. And here's the assembly happening. Golgi's function is the assembly. And then the virus release from the dish cells. And then so a lot of viruses releasing, and then it infects the new cells. We can see the NS1 protein substitution, so it also releases non-structural proteins. So when you measure in the blood, you can see those non-structural proteins are also present. Do you have luck to open this video, or? Yeah, that's what I was thinking to switch my computer. But anyway, so we can show. So move on to the next slide. Okay. So there are 4 serotypes. 1, 2, 3, and 4, and the incubation is 3 to 14 days. What is the incubation? Means once I got the virus. Oops.
Seeking for the reviews.
I'm taking for the videos.
Oh, okay. Shall I wait? Sorry for this interruption. Yeah. It's very pH. So they're making more viruses now. This is finalized development. So thank you.
I have a question.
Yeah.
So, like, whenever some foreign DNA gets foreign DNA or viral genome or something gets into the cells, there will be internal DNA or RNA existing. So, they will not cut the viral genome and make it non-usable for again integrating into the genome and making viral mRNA or proteins.
So it's different, like like mRNA vaccine, right? We're talking mRNA vaccine.
No, no, whatever it is, kind of like when foreign genome comes into the cell.
Right.
So there will be already DNases or RNases will be.
They use from the host.
No, they will not cut the foreign DNA and make it non-usable.
No. Because that's why those they have like pipe cap, zip diamond, everything, and also it cannot be cleaned. So the virus is so smart than a small cell. They don't allow to clean those until macrophage. Macrophage, cytokines of those, they may damage the whole cells and they got destroyed. But normally virus DNA cannot be cleaned so easily. They are very protected. So yeah, like in HIV, there are a lot of studies going on for how to make a drug which leaves those because it has a reverse transcriptase inside, and so it becomes double-stranded RNA. So how to stop those double-stranded RNA? So they have made a drug which actually binding to the reverse transcriptase so that double-stranded does not form. That way they prevent. But as such, if the single-stranded RNA goes into cells, the cell cannot do it. So because those RNA use the host DNTPs and just make copies. And we have the endoplasmic reticulum, and we have a lot of non-structural proteins and all those that actually helping to make a lot of RNAs.
Basically, hijack the host system.
Exactly. So it's a hijacking the cells, and the cell is just sitting and producing something foreign.
So,
Right.
on that, so if the cells that are being targeted are immune cells, how does one recover from dengue? For example, I had dengue when I was a baby, so I recovered from it. Now, since the targeted cells are immune cells, at what point does the reaction?
Some slides coming, yeah.
Okay.
Some of the answer we can give you there, but I'll address it when you don't have some of those.
You can.
So,
Yeah.
how it is relation with the platelet depletion, because platelets are enucleated.
Yeah.
Because dengue is more common. Important symptom is the symptom.
Right.
Depletion of
Drastically
platelets.
reduced.
Because the platelets, what's the function of platelets?
Clotting.
Platelets is a sticky cell, right? It goes somewhere where there is a hemorrhage or there is some inflammation. So what happened when the dengue happen? How are we talking, about the symptoms and all those? When there is inflammation or something, like there is no cytokine. So when we talk about inflammation for the general audience, the inflammation induced by cytokines, chemokines. Okay, those are actually produced by immune cells. So they release. The body defense mechanism is to produce those cytokines. Okay, I have a fever. You can see tons of IL-6, interferon gamma, TNF circulating in my blood. If we do test, blood test. So that is the cell's function because they produce those so that they can reduce or kill those organisms. So platelets function as platelets is sticky, and they say, oh, there are so much cytokine there. They just run there, and just to say, oh, they may need me, right? Same like neutrophils. They move fast. They go neutrophils and platelets there, and then they stay there. They think they can do something, and then you lost the platelets. The platelets function, maybe blood may reduce, maybe it went to the tissue. In different hemorrhagic fever, it's a different scenario. Like, especially because dengue is also another hemorrhagic fever. So they go there. They go inside the blood vessels, but blood, when you take out the blood, you can see, oh, the platelets is low, but the platelets are sticking there because they are not circulating anymore. They are just doing, oh, they may need me, body may need me, I have to help. So that kind, because they are not like a brainless kind of thing. They saw, oh, cytokine is there, I have to run. Same like neutrophils. They go, and it happens.
Next slide. Okay. Is that slide? Okay, so this one. So the viremia, so this is the incubation stage, like 3 to 14 days. So once I got the dengue, some mosquito bite last night in my hotel, maybe. So after I can find out it's 3 to 14 days, right? so average is 4 to 7 days, and that because the virus is, as I said, the virus replication. The cell virus went in and it's trying to multiply now until my symptoms come out. And the dengue fever is always like fever, severe muscle ache, headache, rash. Very difficult to distinguish with the chikungunya, malaria, because all are same kind of symptoms. The severe dengue infection only happens 5%. As you have asked like question in your childhood, got cured. Because you're young, your blood cells is very active, your immune cells are active, you got this. But but severe dengue happens like if I have some other conditions. I have a diabetes, I have other immunocompromised, maybe I have a cancer or something, then I may be very prone to die by dengue because the immune cells are not functioning well. So there are two kind of things: dengue hemorrhagic fever and dengue shock syndrome. So what happened? The dengue viral infection, though, the two kind of asymptomatic and symptomatic. So yes, in childhood you may have a little fever or something, but not too much. No rash or nothing, no headache, maybe just a little fever, and you just take maybe paracetamol or something after prescribed without doing any blood test, and then you're okay. But that at that moment maybe just so we come out the immune function. So maybe the immune cells are very active and just control. Fever or dengue hemorrhagic fever. So there's a plasma leakage. So that point, hemorrhagic fever, when it happens, that's the platelets function is because platelets are sticking to the blood vessels, and that's why you have less platelets in the circulation. So platelets go down. And when there is a plasma leakage happens, there's a very chances you get a shock syndrome. Shock syndrome, that organ failure may happen, and you may die at that point. Or no shock, maybe you can recover that. And the dengue fever syndrome is also something without hemorrhage or with unusual hemorrhage. Maybe normally what kind of hemorrhage you don't see is unusual hemorrhage happening. So that's why they're differentiating the fever and dengue hemorrhagic fever. Hemorrhagic is very, very, you have to be very careful and take proper precautions, go to the hospital or do all the tests and medications. So that is the febrile stage. So initially you can see the green bar showing how the virus, the viremia, the fever is coming, and then inflammatory response is coming a little late, and then it goes plateaus. And the recovery phase, as I said right here. So the immune response, your question. So the immune response comes at that point. If once you have a fever, the immune response comes, and then that controls the virus. Because you have a good macrophage and other cells, cytokines, they release, IgG, IgM, those production happens. And then it will control. So there is the opportunity, if you drugs, an opportunity for antivirus. So you can give some antiviral at point when you have fever, and if you see the, your neighbor has also dengue 2 blocks from your house, most likely you may have a dengue too, because maybe that locality that mosquito is prevalent very much. So you can directly go to the antiviral drugs and treat it before you do all kind of tests. And that's also you can do in the laboratory.
So I have a question here. So without irrespective of intervention, by day 7, the effects will be gone. The symptoms
Yeah,
will be gone.
yeah.
Generally.
Generally.
In most of the population.
Most of the population. It's a healthy, this is the structure of a healthy population.
Okay.
Yeah. But if I have other conditions, other comorbidities, I'm so old, I have diabetes, so it's a totally
It might be complicated.
it's a totally different scenario. I have cancer, it's a totally, I may be dead because my immune cells already depressed. There's no more cells left of the baby. So that's
So without taking medication also it will come down drastically.
yeah. It is normal. That's why I say 5% are let's go to the hemorrhagic shock syndrome. But normally if I have a dengue, I have fever, take something, medicines, some general medicines, and maybe they can, doctor may prescribe antiviral and then go on. So that is that's one of the questions here. Like how the vaccine. So there is a vaccine already on the marketed, the TAC-003. I don't know in India they have a vaccine yet, but how the vaccine works. So you get vaccines. So this is for the chimeric dengue virus derived from dengue 2. So they have what they do with the dengue virus. So there are a lot of envelope proteins. So they check out different parts of the dengue because there are 4 serotypes, and they make a chimeric virus. Okay, this is a chimeric virus, and they use for vaccination. In that vaccinations, you can do the serotype-specific neutralizing antibody as well as CD4, CD8 response. So you are generating good T cell response as well as good antibody B cell response. Okay, whenever I talk CD4, CD8 is a T cell response. Neutralizing antibody comes from B cells. So that is always important, right? We have a childhood who took measles, polio. Everybody, I think, have a polio vaccine, measles vaccine. Who is protecting this neutralizing antibody? If you go today, check with your doctor, I want to see my titer measles, whether I'm getting another measles or not, if there is an outbreak in future. If it is 1:80 titer, you are protected. If it is below, then they'll say, oh, take another measles vaccine because your titer is very low. So that's so with all the vaccines so far, from the discovery of smallpox, antibody gives the protection. We have studied the measles also, the T cells also gives protection, but it's also long term, but the plasma cells, which is actually giving the long term neutralizing, that's come fast. So vaccines, keep in your brain, vaccine always gives a neutralizing antibody, comes from these cells. That's actually giving protections. Some of the cancer patients, they go to anti-CD20 antibodies, so to deplete the B cells. In those patients, maybe you have no titers, or they are very susceptible to any infections. A lot of autoimmune disease to treat those anti-CD20 antibodies. So just keep in your brain, one lesson always remember, like vaccine gives an antibody protection. And for example, dengue also gives a lot of antibodies as well as T cells. So what happens when you get exposed to the wild type virus? Now I am vaccinated. Now I am exposed to a wild type virus. I went to trip Brazil for my vacation in the beach, and I got dengue there. But at least it's protected because the DC, dendritic cells, will come, and because we have antibody already, so the viruses is binding with the antibody because I have the vaccine generated antibody. The vaccine already covered, the virus is already covered with antibody and taken by the DC and kill it. I have a good CD8 response. CD8 produces a lot of killing cycle. We can have a separate lecture on this. CD8 produces so many good cytokines like CD17, MIT1 beta, granzyme P, those are actually killing functions. Like, the armies have a like a AK-47. CD8 has those AK-47 kind of thing. Granzyme B, CD17 and B. So that's the release. And when the disease infected with the virus, it just kill it. Okay, so that's the basic principle of this vaccines. There are a lot of vaccines has been tested, and as I mentioned, like TAC-003, this is very good, and they have been tested in different hospitalized dengues as well as biologically confirmed dengues. They want to see whether the efficacy is good, so it's shown by the green color. No efficacy, possible enhanced, or possible enhancements. The vaccine is enhancing more infection or not. At least TAC-003, there is no enhancement, so the vaccine is more safer. And unknown efficacy means still unknown, because it's just the vaccine came out, so they still need more research to find out if there is no answer. So please run this video. So this is interesting. So because there is a question like, I have a dengue, I recovered from that. Okay, last year maybe. It's a hypothetical. Okay, I got dengue and I recovered. This year also I get dengue and it's very severe. Why it is so? That's the video to talk about. Because what happened, the antibody, last year I have a dengue, I got a lot of antibodies, right? Neutralizing antibody and it protected. But no neutralizing antibody actually helping the virus to replicate more. And that is the bad part of dengue, and which is not for any other viruses. And that's why when you got the second infection, the dengue may be very severe. It's sometimes life-threatening. So the AD is antibody-dependent enhancement. So those antibody which is produced from the first infection enhances those infection further. So you can get an idea from those.
Welcome to the research services.
Right. Infection of one. ADE. Yeah.
So do you say there is no end for the ADE? Like as and when there is a new so
There is no way we can
various
stop
antibodies.
ADE. There is no way. That's why the more emphasis should be given to make vaccine at least. So I saw the other slide. If you have a good vaccine, like the TAC 003, which is very promising. I don't know if India is available, but that can protect ADE because that does not generate any more ADE. If there is one thing comes, it controls that.
Does it work against all the serotypes of
Yes, it's a chimeric. So chimeric means it has all 4 types. And they tested.
Some type is here also it's available. Don't know that exactly what they say. Vaccine is available.
No, vaccine is that you can make the vaccines like inactivated those things, but that vaccine which is tested, then yeah, because here you can say take the dengue 1 vaccine, but it will not protect 2 and 3 and 4. But that chimeric one protect all of them. So that is a good.
Is there a difference between the response that you would get from the antibodies that are produced from the vaccine and by our cells when you recover? Let's say I get serotype 1 and I recover from that.
You have an
And-
antibody from the serotype 1.
Yeah, so that might help, or that might cause ADE for 2 and 3. So
No, that may help you for only A, but it may cause ADE for 2, 3, 4.
2, 3, and 4.
Exactly.
So is there a difference that my body creating antibodies would, like for example, I take the vaccine that has all 4 serotypes, the chimeric
Yeah.
vaccine.
Yeah.
So that would protect me from all 4.
Right.
If I take a vaccine that is only protecting me from one serotype, would that help, or would that cause ADE for the other serotype?
That cause ADE for other serotypes. Yeah. Because you are generating, you are generating for antibodies. So there are some vaccines they already tested, like only for one serotype. It is not good for that. So
it's only useful if it's all
only
4.
for that serotype. So that is the lacking. So that's why dengue may be discovered long ago. Still, people are struggling to make a very good vaccines, and that is the idea behind it. So to get a better vaccines which can cover it. So yeah. So I think this is the end of the story. Let's move to the short one, or maybe the second disease emergence. Actually, I should start that first. So giving an overview of what is the disease emergence, so it is the identification of the etiology of a well-recognized human disease. Sometimes working, not sometimes not. So I have to point here or that side. Here. I don't see any effect of this. But yeah, just go on. Next, please. Yeah, so the increases in human disease caused by an extincting pathogens, which increased. It's a kind of general, like you might have given before dengue. So it's increased the host susceptibility, increased the virulence. Next, please. Accumulation of susceptible host and pathogen reemergence. Next. Pathogens new to specific human population. So that is the emergence. Novel pathogen. So when you say, oh, there's a disease, there's a pathogen emergence, so it falls all these categories, okay? So there may be new disease, it may be affecting the new human populations into the specific population, maybe it was before only African Americans. Now I can see that diseases in Asia too. So those are the things. That is the difference. Next slide, please. Next. So these are the the spatial distribution of the virus species. You can see the small circle to big circle. There are how many species? As it increases, the circle becomes bigger, and we are looking all those continents. Okay, and you can see those species. You can see big blobs like here. India. So there are lot of species are generating. Like this is the curve. Why it is happening? This is the curve. It's not clear here, but as the year progresses, and this is the viral species. So the earlier very few species were there, and you can see the trend it's increasing. That's the point of this slide. The point of this slide is, as the days we are progressing, there are more species are generating for single bond at species level. Whether for dengue or like HIV, I will talk about HIV later. There are more species evolution happening. So the virus, what is the virus? There's virus, it's called like flu, reassortment happening. And there are a lot of different serotypes happening. HIV also, there are a lot of mutations happening. There are new mutations, COVID, initially 2020, there are a few variants, and now you can see a huge list of different kind of mutant various viruses are. So that means the species are increasing. The plot is getting bigger and bigger. So that's happened. And that is the normal. That is normal for viruses. When virus gets in, it needs a patient. It needs a person. It needs a human where they can make those mutations. Virus cannot do anything in the floor or in the board or in the microscope anywhere. It needs a live person, especially human. And that's the happening. And so it can correlate with the human population also, because human population is increasing. Virus diversity is increasing because they need the humans to multiply and make different mutations and all this. Next slide, please. And so this is kind of a general overview how different. So here categorized based on A, B, C, D. A, you can see like how many helminths, how many different fungi, protozoa, virus, and bacteria. So the bacteria is always high, heavy level. But you can see that as the different years goes, 1940, but their numbers is increasing. Okay, emerging infectious disease, EID events, EID for emerging infectious. So there, you can see there is a trend. All the plots, helminths increasing, parasites increasing, virus increasing, bacteria increasing, everything is increasing, as well as the number of defects increasing. Also, the drug resistance is also increasing. The vector-borne is increasing. So there is always trend, and because the human race is increasing, virus mutation happening, and all those happening. Next slide, please.
This increase in number, it could be due to better diagnosis, better detection, and improvement in the kind of detection.
So your question is the detection?
Yeah, it's because of.
Of course. They are correlated too. Because earlier 1950s, who knows, I have a PCR, right? That time there is no, and there's no infection, but you saw that like a plastic plague and all those plagues happened, Yersinia pestis, those happened long ago. Even that time tuberculosis were there. People don't know it as tuberculosis. So that is absolutely correct. And but still, those graphs have been made based on archived samples. Like the 1918s people still have. I'll show you one picture. 1918s still people have the sample from tissue samples where they first find out, okay, this was ASN flu and what kind of flu type. That time flu people don't know, it's a H1N1 or H5N2, right? That came recently, maybe within 10 years, because people had those tissues, archived tissues. And then they find out, okay, what type it is, what kind of flu it is. So that data is there, and those informations
People do archive, people go and dig into a body, mummies and all those, they can collect samples, and then they find out why that death happens. So that's another scientific discovery there. So there's
Some of
the,
this is also related to climatic changes.
of course, yeah. So I'll cover that maybe 4 or 5 days. So yeah, definitely that's a very valid point. Climate change, human traveling, as human are traveling, vectors are traveling. Why suddenly dengue is endemic? No, it should not be there. Aedes was not there, but now West Nile is there because Aedes mosquito is there. So yeah, of course, unknowingly, knowingly, people carry luggage because there may be a larvae. Who knows? Who cares, right? A mosquito is there in your luggage, and it came out. That happens. So many ways. Even the trucks, forget about the flights. In huge cargo ships, lot of transportation happening throughout the world, one place to another. Bird migrates, and I'll also talk about how bats we know bats live for longer, and bats can travel so many miles distance. And yeah, that is always there. I mean, that's also related to the humidity, the temperature, because the mosquitoes love more humid, more temperature. That's why it's happening. The climate change is happening. Ice is melting in Antarctica, so those definitely has a huge impact on this. So this is another slide so showing that how the major emerging diseases for the past 20 years. We saw so many disease here. I'm not naming everyone, but some of them like the monkeypox. We heard the term monkeypox recently. There's a huge outbreak after the COVID. I think it's like 2022. 2020 was the COVID one, and then 2020 is the Guinea. It's not important. It's only the Vietnam happened, but 2022 the monkeypox as a pandemic. It was too much in USA, Africa, even there's one death in India too. And
Western Ghats. Western Ghats.
Yeah. So that's the kind of different scenario. So this is another like a different way to see how total infected cases so far, and here the C is how many deaths from the major, and then weekly confirmed COVID-19. So that time everybody is watching how many deaths happened COVID-19. No, I don't think anybody watched that, but that time it was a very it's a human psychology, right? I should not get infected, right? Or if I get, what will happen to me? What will my family? So, I mean, that's the true sense. I mean, it is normal because there's that time there's no vaccines. We don't know, and a lot of information is unknown. So that's the scary thing, and that's why this topic, what we are talking today, emerging universe. So that's very important to know. At least prepare yourself for the next. If something happens, what I can do. Even unknown things happen, what I can do. So you can see that a lot of death is the major COVID-19 happens. This is pandemic. We had the Zika infections. I think in India also 1 or 2 maybe cases in Zika, which is mostly affects the babies, which are pregnant mothers. It is a huge virus in Brazil, that's Southern Africa, Southern America. But not too much other countries. The monkeypox also now it has a good vaccine, so it is okay. So this is the kind of
The concentration of the disease in a specific area, is it going to play with respect to immunity of the people living in that area?
Yeah, of course. Yeah, because some of the disease go to human transmission, right? Like direct contact. So those are the like COVID. It is not by vector. It is by the direct contact. If I touch it, I have a COVID, I touch it and you touch it, so there's more chance by you got the virus. Or through the aerosols or droplets, if you're talking and sneezing and those things. Like TB, TB is the oldest disease. It's still India has a lot of TB cases, tuberculosis. If I travel a bus, with somebody coughing, I don't know that guy has a TB and I may get TB too. Do we take appropriate precautions? Have a mask? No.
So I think that's I think I'm looking that direction. Like the new generation and new people should take precaution, and that's the way we can control. I think if we don't be careful enough, I think it will still go along, and people like me, some people will come and lecture on it. I mean, there will be no cure. I think that's why the importance of the education is like, we should be aware all the time. Even if we go outside, we always be suspicious, like, oh, maybe this person has something, I should protect myself because I will go home, my old parent, old mother is there, old father is there, they may get infected because they are very susceptible, they are already older. So already aged people has more susceptible than younger people. So those are the things. So this is a summary, and also this is the book. I have a book chapter on this. We published recently. So how many deaths so far? The HIV is the major, which is my major research for now. 40 million deaths globally happen. That is still, pandemic. It's still happening, but not get too much attention because we'll talk HIV more on this. HCV, hepatitis B, it's always there. Hepatitis B has a good vaccine, but still hepatitis B is global. So that's another thing. We are not aware enough. I think it's more comes from the awareness of the human being, how you can control. Something is very easily can control by generating awareness. Monkeypox only 970, and influenza A, it's a major like 650,000 globally. So this is the.
So what are the chances of vital genetic material getting recombined with human? Determinants, or what are the chances? How recombination?
So you are asking to?
Many viruses.
Yeah, like SARS has a mutation in different way. It has some recombination, but it has some more mutations on the spike proteins. Okay? The spike is more important. Like all the vaccines developed because the spike mostly focusing on it. But influenza has more reassortment happens because we'll talk maybe tomorrow. How their hemagglutinin and neuraminidase, how they change, and for that it's important to make it reassortment or the recombination happen. Like I got influenza A H1N1 2 weeks ago, but this is the very mild one, and I never felt any fever or any coughing, nothing happened. Yesterday I was eating Billy, and I got somebody was there, sneezed too much, and I got H5N9. It's also mild, right? So nothing happened. But in our body, two viruses came and they are making reassortment. And now I got like H5N2, which is very pathogenic, and I have, after a couple of days, I have to be hospitalized.
These mutations are carried through generations?
No, not necessarily. No. It depends on the person. Like, because the virus only needs that person. Like, I need Bucky, to make new viruses. And once the virus release, virus needs always some living cells. Otherwise, virus is dead. You were saying like how it was genetically transmitted. It only happened when it is a mother which is pregnant and it transmits to the baby. Especially happen when HIV, HCV, hepatitis B, they may transmit to the babies. So that is kind of, we say, like genetically transmitted to the babies, and the babies got infected. Zika virus also, like mother got infected with Zika, and then baby born, he has a bigger head, encephalopathy, and encephalitis, those things happen. So, yeah, but not all the viruses follow the same pattern. So this is the process of pathogen spillover. So the spillover is very important. Like how pathogen spillover, like one of the questions is saying, like the climate change, right? So the pathogens, like this is the evolution happening within the bats and different wild animals in the forest. Okay, we don't know that. But I went to the forest yesterday to explore. Oh, I never went to the forest. Maybe I will shoot a deer there. And I went there, but somehow a bat has some excreta or urine, I touched it in the forest, I don't know. Okay, and I touched my mouth or something, so I got the virus came from the bat. And then I went back to my home and I transmitted the same to my parents. So that means the spillover happened there from the animals because I visited the forest without proper precautions. And I got touched with those and I transmitted to the urban area. So the spillover is happening for long because there's a, and also there's an intermediate host also plays role here. So they are like pigs, chicken, we don't strain all those, right? They are reservoirs. How we are cooking the meats, is the meat is well cooked. Those are non-vegetarian, there's no pathogens left. Those kind of things. Some people use raw, Japanese, like sushi and all those, raw meat, raw fish. It's a tradition, different cultures. So that's the way also spillover happens, right? How you take the COVID also they say that it's generated from the laboratory, or it is like a bad culture things, people are eating those bats or other things which is transmitted from the bat, right? So those are the questions we cannot answer until there is enough funding research. So the spillover is always there between, there are a lot of virus which is we don't know at all. We know maybe 2% or 5%, but there's tons of bacteria. So that's normal, right? Parasites, bacteria, helminths, protozoa should be there. But how they come to the human, maybe they are not pathogenic for them. But once they come to the human, they have like a recombination, mutations, and they become highly pathogenic. So this is the important I like to discuss. So there are a lot of WHO, World Health Organization 2024, they have this is the priority. These are the very important, high priority public health interest, public health emergence of international concerns. So those viruses they have reported in 2024, and people are heavily throughout the country of different Europe, Asia, America. They're following this, so if there is any outbreak from those pathogens. So I'll be talking in this week some of the viruses, it's not possible to cover everything, but some of the important viruses, what we know so far, how they transmit, what are the precautions that's been taken, and their future of it. And, so, some of the viruses, also I keep on emphasis based on Indian context, what India, what are the viruses are more prevalent, and also kind of emerging viruses in India in the coming years. And not reading everything, factors that lead to the emergence, as you questions like, poverty, economic social development, society background, human factors, biological factors, cultures. So these all play important role in our emerging diseases. Some of them increase global travel, and as you say, global warming. So climate change, global warming. So those all factors because cutting on trees where the animals will go. They are hiding in our house. Oh, this is at least some place to live in. So transmission happening there, and then rat is another one, very important transmission vector that carries a lot of pathogens for the humans. And the rat eating your food in the night, and you don't know, and you get consumed next day, you may get infected. But rat has before. So that's all the list. Poverty is another. One of the major is like a lack of safe water. A lot of we always see in the newspaper the food poisoning and all those because we don't have a safe water. That's another thing. Societal breakdowns. We all know war. Human factors, sexual activity. How people are doing, entrepreneurial users. Substance use, people consumes, a lot of addiction and all those may control the cultures. cultures is vaccine resistance. You probably heard that Western countries, there are a group of people who are reluctant to take vaccines. They think it's bad. Sometimes they put in a religious aspect, or Justice Christ has not said that you can do. So there is a lot of controversy. Then you have to also um, respect their cultures too. I mean, you cannot culture is very sensitive thing. So yeah.
So US Health Secretary Kennedy, he refuses to believe that vaccines really help. He doesn't believe in vaccines. Even Trump doesn't believe in vaccines. So does it really affect the entire nation's program of health?
He never stopped. I mean, whatever vaccine, like if you go to the CDC website.
But if the main person thinks that the other way around, then it's a problem. Like he's the one.
Yeah. So there's still discussions. People believe that way. I think the latest news so far I know is that Jane Kennedy said there is no change in the vaccine regimen for the children. But there are some vaccines like MMR, like measles, mumps, and rubella. There has been recommendation that you separate those vaccines. Measles separate, measles, mumps separate, rubella separate, so that they can get better immune response. That's I heard, but there is no such concrete discussion on the development. I don't know. So mostly the CDC people, folks.
Yeah, so because it will kind of like create a situation wherein like the administration itself is not interested in the vaccine development because they are not believing in that. So
No. There are people. I mean, still developing vaccine. I mean, even J. Kennedy.
Because government funding is needed for most of the.
Right. Yeah, there is a still interest to make vaccines for lot of other diseases like we mentioned, like Nipah. Still they're funding because Nipah is another we worked recently. Nipah virus, enterovirus A71. There is no vaccine. There is a huge interest on this. And female congo, so this is also very prevalent in India. Female congo, there's a lot of studies. So yes, there's a lot of research on those aspects. Measles, it's a controversy. But measles, we know all measles are very good vaccines. It cured a lot. I have some slides to share with you later. So what is eradication? I mean, control and elimination. So this is kind of like control means we are just controlling. Disease is there, but you just control it, right? Elimination of disease means there's zero, nothing is there. Like smallpox, right? We totally eradicate. India is eradicated smallpox. There is no case of smallpox. And elimination of infection. So the disease elimination and infection elimination is a little different. It's a reduction. There is no incidence happening, but I cannot assure there is no disease. Maybe the disease is somewhere inside some animals or somewhere hiding, but I don't see any infectious disease. Extinction means no longer exist. So indicators of important medicality. So these are the again common things like effective interventions. How it's a very generic things. How we can stop the irritability. So those are the effective interventions to interrupt transmission. So how, for example, dengue, how we can take measures to transmit from mosquito to human bite. Mosquito net, one spray to kill all these mosquitoes, or use some anti-mosquito something, dust or spray. So those kind of measures you just take. Practical diagnostic tool, sufficient sensitivity, we need good sensitivity assays, right? Every assay has limitations and also the price, right? Maybe PCR like COVID, for example, Apple was so busy to say, hey, whether COVID or not, especially I know when I was traveling, I have to show the airport like I'm COVID negatives by PCR. That's the only test they accept internationally. So I have to go to them and they get, I'm PCR negative. That's then they will accept and they will help me, allow me to travel. So that's kind of thing. Like now question comes, okay, tell me your PCR, how much sensitivity. Do we discuss that? Or shall I write in the report section how much sensitivity your PCR is? Probably not. Is it all the Apollo Hospital or others report? Maybe not. So, some lab has very specialized, like they can detect one single RNA, one single virus. But for that you need very lot of volume of blood, lot of testings, make sure your target, your primers are well designed and do the sensitivity. It's not like one test you do and done. You have to do multiple, nested PCR or some other PCRs and do different. I'll talk about end of the day maybe. I think they tried what are different assays PCR people have developed so far to define those populations. So that's the other thing. Right? Sensitivity is a very key. We do ELISA too, sensor. And humans are the only, so not an absolute variant, but it's more complicated. So as you know, humans, we, our immune system different. They are two sitting together, but their immune system is different. So they are both gets dengue, one can recover faster than the other. So what is the reason? So because their genetic makeup, they need to study. That's why the major field came precision medicine, right? People, in America is very popular nowadays. Like people who are have a more money there, they do their own genotyping already, and they find out, okay, I may be prone to breast cancer or cervical cancer or this, so I have to take precaution only. I'll not eat red meats. I'll not eat those. So this is called precision medicine. So that's in Western culture, it's very popular nowadays. So that is the another thing. So even those two ladies sitting here, one genotype is different than the other. Then one person may be susceptible to one pathogen, maybe the other. Maybe the other one is unsusceptible to the other. So those are the another things. It's important. So prime importance and challenges of sustained political will, as we all know, we have no need to discuss how the politics and political situation in every country plays a role to take the decisions on what is right or wrong, when to use mask or when not to use mask, when to take vaccine, or not to take vaccines. Even for HIV, I like to highlight, like in HIV, there are a lot of people who are still infected, especially in Africa. I don't know about India, I'm not gone too much into say. But there is a very good drugs available for HIV. But is that everybody has the access to take the drugs? No. Then the question, why not? Political issues. Maybe they don't want to spend the money, or they have some other issues, or So okay, so I'm done with this slide. So maybe I can take another, maybe HIV. Okay.
How long this would go?
You said 1:30, right?
Yes.
I'll try to finish one. That's why I'm watching every time. Like,
No.
So
This is
anybody had never heard of HIV? That's my best field of subject, right? Because I studied a lot. So human immunodeficiency virus. It came 1981. I think the first report has been in Los Angeles. Two guys. It's a I think I can fairly talk those.
Yes.
Was homosexual and heterosexual, right? So guys, homosexual, like gay, lesbian, and those heterosexual, like married couples. So it is very common in 1981. There are a lot of homosexuals, especially gays. They find out fast that the disease, they are weak and all those. So then they find out what is the virus causing, and then they find out it is HIV. Next slide, please. Okay. So number of people living with HIV still now it's 40.8 people living infected. So every year you can see, for example, 2024, 1.3 million. What that says, 1981 we got the virus, we saw the virus, it is well established. Everybody knows HIV. Still 2024 we are getting 1.3 million infections. it means we are not sincere. We don't take it seriously. I mean, HIV can ruin your life. Well, now we have drugs, we can still live for lifespan, but still, nobody wants like taking those drugs and having side effect and all those. And also, there is a cultural taboo still present in some places, right? People have HIV. They don't. There's a lot of. I think you should watch that movie. Tom Hanks, very used to like Oscar. It's about HIV, how that guy was. I think.
Philadelphia?
Yeah, it's Philadelphia. It's nice. You saw that, right? So the Philadelphia, I saw that it's a very nice story about HIV. How that guy has been taken out his job because they find out this guy, Tom Hanks has HIV. He's the character. That character has HIV, and then he fight for that in the court, and finally he win. But he died because that time there is no antiretroviral therapy and all those. So the point is the negligence, right? Still in 2024, we have 1.3 million. It's a big number. And see death every year, 630,000. The more important is see this number. Number of people antiretroviral therapy as of end 2024, June, 31.6. So it's all subtract 40.8 minus 31.6. 9.2 million still without antiretroviral therapy. Then the question comes, we have antiretroviral therapy, why 9.8 million or 9.2 million people still don't have the access to those drugs? Is that political? To some extent, is that poverty? They don't have to, cannot buy those drugs. I mean, that's also political drive. They don't want that the citizens should be rich or taking their healthcare. So definitely there is a lack of healthcare in those places. I mean, it's more majorly happening in the in the African regions. And this is the kind of distribution. So, and you can see in Asia, I like to highlight Asia, which includes India too. Like 23% of the HIV infections happen in Asia also. So India is one of them. You can see there is a significant progress in the response to World Health Organizations. They are using the drugs, and that is the target. HIV infection should go down, and number of AIDS related should go down. But 2025, we should be here. That line should touch here. Is that line touching here? So that means they're not in the mark. What they predicted they should do in 2025, how much prevention should be done, they're far behind. You see, that much gap is there. So AIDS, as I mentioned, it's 1981. All the respective studies suggest that it occurs. That's people recommend like, oh, it happens, and the virus. There's a famous, HIV biologist which I've worked with, Preston Barnes, who worked a lot on this. And he has a theory that it mostly came from monkeys. Monkey has a virus for long, and they can survive without any death, or so the virus multiplies. And somehow in Africa, the people eat monkey meat, the butchers, and they sell it. And somehow when transmitted to the human, in consumption of the meats or the blood or something, and then it has mutation happens, and then the HIV has to come up.
So same thing like whatever is affecting humans, causing severe disease or whatever it is. So that doesn't cause any disease to the monkeys who were posed before this.
Yeah, posed to HIV does not affect the animals.
Even the changed one, whatever.
Even the changed one. But the original one, the monkey version one, even though the monkeys are not dead, those are. Okay. But they have a virus like bat. If you can take a bat and study, I will show you one slide. There is a huge consortium going on. They are trying to do screening of so many species of bats. I think the project will end 2030 30 or 40. They are sequencing and making sure what the bat has, what the bat different organism has, because a lot of disease transmitted through bat. So yeah, I mean, in respect of HIV, monkey has the HIV for long. So that's they say 1930, but it's still not there.
But replication do happen there involved.
Huh?
Replication, multiplication happens in the
Multiplication happening. Continuous, they're shedding the wires, but they are not affecting their
Oh,
health.
we
Yeah.
have this thing not confident.
So then, yeah, I'll move quickly. 10 minutes I have. So the M and O, there's so many subtypes here. There, so the major is the HIV1 subtype B, which is that's kind of remembering like Europe and America. So Asian, and this is very important. And HIV1 subtype C is the mostly they have it. And this is kind of subtypes in a distribution of those. You can see the in India right here. It has both B, C, and mostly B, C, and CRF. So can you play this? So I'm giving like a very nice video here. It's just great. They can watch through YouTube too. Yeah, so go back to my lecture. So these are the three people got Nobels for getting the HIV discovery. This lady came to the lab when we have a symposium on
So why is about the valve did not get?
and there is a controversy that he found the first these three persons found
There was a
this-
huge controversy.
Yeah, exactly. So he was very close to get the Nobel, but I think I don't know too much on those, but they recommend these three people are the first initial, and Gallo maybe like next year or something, the NIH just find out something. So the credit goes to God first. So. Next slide, please. So yeah, so it's a basic like different viruses. You need to remember that the point is the retroviral family is enveloped it's a positive 10 RNA. The more whether it's a retrovirus or lentivirus, they say lenti means low, and it's a long incubation time. That's why it's not like dengue 3 to 7 days. We can find out that puppy has dengue, but it takes years to find out puppy has HIV. So that's why it's a slow killer, like cigarette smoking or people know drugs, drinking and alcohol. So that's why it's called slow. That's why it's like next slide. Okay, I got it. So yeah, I mean I'm skipping due to time. This is the major opportunistic infection. It happens very late, but sometimes it may come early. So a lot of fungal infection, candidiasis, here leukoplakia, and Kaposi sarcoma, and also you can see the lung. Uh, there is a pneumonia. You can see the fluids are everywhere. The clinical manifestations, all those candidiasis, all secondary infection happens. What candida we have, body, all those organisms have, but because it suppresses the immune response, all come like pathogen. People die due to candidiasis because in normal human being we have candida all the time. I think everybody has some form, which is mild, which is less pathogenic. As we know, like on our gut, several billions bacteria is there. They carry all the time. That's why you take yogurt, maybe some other thing. People take probiotics, prebiotics, because gut health is important. It makes the bacteria healthy bacteria. When it's a bad bacteria comes like Helicobacter pylori and others, it's bad for the same like candida. Good candida is very important, like fungus. We have a lot of fungus, lot of viruses in our gut. We always constantly keeping those. But those may fail after we have immune system go down. So that's happening for HIV too. And those lot of infections come. So the major is the sexual contact. Sexual transmission is the major. It's also having vertical transmission, like from mother to child, and blood products. There's a lot of cases happen in different countries when there is no screening facilities. Whether I need A1, A plus, and somebody O negative, then there's no such screening whether the people giving the blood is HIV negative. Now we have HIV, hepatitis B negative, hepatitis C negative, those things coming up. But earlier there is no screening. Just take somebody and put the blood. So I think everybody knows like Amitabh Bachchan also got the hepatitis B because that time there is no screening. And he lost his liver also, like part of the liver is gone. So I can't confirm it. But it's been mentioned because it's a blood transfusion. So HIV also happens to the blood transfusion. So this is the important slide. I like to say how the transmission happens. The oral sex is very low. The people who have those some small risk with the oral sex. The needle stakes is 0.03%. The transmission with the sharing needle, which is 0.67. So it is loaded. The maximum happens is the anal intercourse, which is about 1 to 30%, and vaginal intercourse is less because of why cause what is the reason is because in a vaginal environment it is a mucosal tissues. There are a lot of fluids, a lot of cytokines and other things there. So which actually protects because the major cells are which are the target cells, the CD4 cells. Okay, CD4 cells sometimes macrophage. Those are two cells, and then it needs receptors CD4 or CCL5, then the receptors. And those cells in the like a vagina, if you look up the vaginal anatomy and anal anatomy, the cell concentration is similar. But the vaginal mucosal environment has a lot of different mucus secretion in the vaginal fluid, which is a lot of antiviral, antimicrobial cytokine chemokines present. But anal is kind of more like a not that much. And that's why there's more transmission happens through the anal intercourse at this. So this is a kind of schematic, the presentation of how different transmission. When he was 20 years old, he discovered the Langerhans cells, but he died due to TB. I'll skip that. So basically, the stages of infection. When you get an infection, there is seroconversion, and you can see the time. It's not days; it's months. And you get some antibody response. And then clinical symptoms, asymptomatic HIV, 1 to 10 years. That's why it's a longer. If you get infection, you don't know. And then and the P24, which is that's why the seroconversion. So you can see more IgE, P27 antigens, and antibody, GP130 antibody is picking up. CD4, the red one, CD4 is going down, and then. At the later stage, like it is like a several years after months, you can see the virus replicating again because the CD4 is going down. So the cells that respond to HIV infection are the same cells that HIV infects and destroys. So it's very kind of paradox here. Because the cells, when it infects, it actually kills the same cell. And it causes immune suppression. So there is a lot of dilemma. Earlier, the CDC has recommended CDC is the major. I mean, who ever never heard about CDC? So Central Disease Control, which is a major health organizations in the United States, and WHO is one of the organizations. So they have two different criteria of defining HIV earlier. I think they are in the same format. So earlier they have one person was, I think WHO is saying percentage of CD4 depletions. CDC is saying no, it's the absolute count, how much CD4 actually counting, how much cells are going down. So the 50 cells per year, if it is decreasing, that kind of thing. So they said. So this is again another picture you can see. This is the red one, is the viral load. And this is the RNA viral is replicating high, then it's go down because then some antibody response come here, and then when the suppression goes and thing happens. There are tests available, ELISA, Western blot, which is an antibody detection test, like people do for job purpose. I know when I went to DRDO, I have to give the HIV test to make sure that I'm HIV negative. So they do just ELISA. They do ELISA test and show, I don't have any cell conversion. I know cell conversion. If I get an infection, I get the cell conversion. Cell conversion means I should have IgG, anti-HIV IgG. They can detect by ELISA, then okay, I may have got the infection somewhere. Then I should be on something. Maybe I'm dead by now. But ELISA, so that is another test. Most of the jobs, especially defense related, they have to do those type of tests. Even in IH, I have to get the HIV test too. RT-PCR is the more sensitive. It detects how much copies of presence. And there are recent also like rapid tests, which is called like a lateral flow test. Like everybody knows like COVID test, right? Taking sit down and do this. So lateral flow test is also very common nowadays. So these are the symptoms stage, and they categorized WHO 1, 2, 3, 4. 3 means very advanced stage. 4 means we have AIDS. So it's very difficult who has already 4 to survive that point. So as I was mentioning, like the CDC, they always say how many cells. It's the absolute CD4 count. But WHO never said that. WHO says like a percentage. Okay. So this is the natural history, and you can see there are a lot of how the CD4. So this is very nice. Like what opportunity infection is going. Like as my CD4 is going down, you may gain those kind of infections. All those as you go down, you can so the CD4 going down, and you get more of those. I mentioned like Kaposi sarcoma and sinusitis, tinea, so those kind of all symptoms will play out. Treatment. Next time we'll make sure that's
Yes.
so the treatment is very nice. So there, I like to highlight that in September 2025, CDC has recommended us lenacapavir. That's the drug which you can take every 6 months. I think so. Every 6 months, just one pill. Earlier, you have to take multiple pills. HIV, like if I'm HIV, you have to take every day so many pills to suppress the viral load. But now this is a very nice by GIAN. It's a very famous company in California area. They discovered so many, and this is one of the inventions they did, lenacapavir. Every 6 months you take and then forget about it, you're not take anymore. And it controls the viral replications. But
What is the target for this drug?
Capsid,
yeah, it's a capsid inhibitor.
cap protein.
Yeah,
Capsid.
capsid. It's inside in nucleic acid, it's inside the capsid. So it prevents the nucleic acid formations. So it breaks the capsid production.
So the virus will not be natural virus.
Exactly. Yeah. There is no weak capsid, so it will not be non-functional. It cannot infect on the
Interfere with the synthesis, but capsid formation.
Right.
Packaging will be
Packaging.
Packaging will not be. So, but these are the general. So one time I was working with the monkeys, and by mistake I put my finger with a needle. I was working with SIV, simian immunodeficiency virus, which is similar like HIV, and our protocol from the CDC that I had to take those drugs. So I had to take.
But it will stay in the body for around 6 months. Right?
But oh yeah, those drugs, yeah, that's why it is a remarkable. They are now working for once a year. Yeah, so that you forget just take January 1st this year, and then wait for the next
But
year.
it is not getting eliminated from the body, then it's going to stay. It's going to be.
Well, body, that is a different question. So, if you get infection, the virus you cannot eliminate. That's why I showed the slide. Elimination from infection, elimination from the disease. Those are two different. Elimination from infection is not possible there. Because the virus is staying in the spinal lymph node. We have lymph node, it's staying there like forever.
Now, talking about drug actually.
Oh, the drug?
Yeah.
The drugs probably somewhere. Because it's also slow releasing drug. You take it and slowly it's releasing. Like a lot of now drugs are slow releasing.
Yeah.
So it does that way. But the virus also is there. It's not like you suppress it, does not mean I'm totally cured. The virus is still there. And if you don't take the drug after 6 months, it will come back. So yeah, so those are there. And so the last slide here. So I think a lot of people have seen these videos, like U equal to U. Like what I also emphasize, like undetectable equals to untransmissible. So that means there are a lot of there are nice videos I actually watched a couple of months ago. So there is a couple that the husband has got somehow HIV, and then but the couple really wants to get married and have kids, but that lady was reluctant to get married because they saw you have HIV, then it may goes to the baby, and baby got infected, and nobody wants that. So then the point is here, if you take the drugs and if you cannot detect the virus in the blood, you're not transmitting also. So it will not be transmitted to the baby if it is coming from the male or female, whoever is infected. If you take the drugs and they cannot detect the virus, it will not be transmitted transorally or genetically to the baby's body. So the HIV, the baby will be free from it. So that's effective. And there are a lot of drugs now, starting from measurements that that's why 1.3 million still people get infected. So there's a simple precautions there, but there are a lot of drugs on the market, which is no, so, yeah. So those are the kind of things, but still HIV is there.
Does undetectable mean cured?
No, as I say, undetectable, the virus is always there. If I'm HIV and I'm taking the drugs, I if you take the blood and test the virus load, the PCR come was negative. That's what means I'm negative. What I say, like the virus is not present in the circulation, but the virus is present in the lymph node. So lymph node is another lymphoid organs. Gut is another major lymphoid organs. So the virus stay there. We have studied I can cite my paper. We have looked over every organs, starting from brain, hypothalamus, thalamus, different organs. We have seen the virus is present, but the virus is not in circulation. Neither it virus transmitted through semen or other things. Okay, so that's why it says inconclude, so it is not transmittable. But the virus is present. If you take out the lymph node for 2 biopsies and check out, okay, what is the virus? It's still there. But the person with ERT can spend their normal life, and yeah, a little over that. So yeah, that's the point.
So with the present available drugs, so how long will the survival last?
Still normal life.
So people can lead normal life with all the
65, 70, yeah. If somebody had been diagnosed early and detected early, like if somebody have this virus and they have detected, early identification is more important also. Like I'm sitting there, but I'm not telling the doctor, I'm not going to them. It's a different thing. But if I say, I did something, I got the virus somehow, and then I start treatment immediately, a good life.
It's going to play with the viral load in the all infection.
Before major damage happens, so if you take it, you will have a normal life.
What is the progress with respect to?
And first of all, another thing, there is no vaccine HIV.
Yeah.
This is still re-emerging virus, re-emerging disease, and that's the point I included here.
I wanted to ask actually, what is the progress with respect to vaccine development in this?
I think it's 0. The vaccine response, there are some monoclonal antibody treatments, like COVID, they use a monoclonal antibody, Trump took the monoclonal antibody. Monoclonal antibody is there, but it's still, it's several like phase 1, phase 2, then it stopped because it's not effective enough. Even the vaccine also, phase 1 went, stopped, because it's not good.
It's shirts.
Right.
Any questions for that?
Any questions?
I want to ask.
You can always discuss with me informally as well. Thank you, Dr. Bobby, wonderful session that we have had about this virus. So we'll break for lunch and we'll gather maybe after 40 minutes.